This is the group. Let's get this one. This is the group that's currently involved in the project, and I'd like to make. There's a wider group that was involved in the diagnostic phase, and I'll acknowledge those at the end. I'd like to make special mention of Lucy lending her great knowledge and skills from the plant side in molecular to tackling an animal disease, and that's a great spirit of cooperation. So probably the first question is, why would we be interested in diseases in crocs? Well, crocodiles, there's only a handful of crocodile farms in the territory, but they are expanding in numbers and they are quite substantial numbers. There's over 100,000 animals being reared at the moment. The largest farm, Porosis, has got about 40,000 and it's increasing in numbers. And while the larger ones, these are probably 2 to 3 year old ones here, getting close to harvesting, while they're fairly resistant to disease, the little ones, which they call the hatchlings or the juveniles, can be quite susceptible to disease outbreaks under certain conditions, particularly conditions of stress. So the background to this project is that in the middle of last year, the crocodile farms, a couple of the crocodile farms experienced a disease they hadn't seen before, and with losses that they'd never encountered before in the history of crocodile farming. And the particular two farms that were affected lost a high percentage of their juveniles, 96% on one farm and 29% on the other, which accounted for over 3,000 deaths in these juveniles. Two other farms just had low morbidity, a few odd cases of a similar disease syndrome in older animals. The investigation to find out the cause, during this investigation, over this 3-month period, we autopsied 60 animals, and they had fairly consistent lesions within this group of animals. They had a severe conjunctivitis combined with a fibrinous oropharyngitis. That's inflammation at the back of the throat with build-up of this white fibrin, which I'll show in a minute. And occasional animals had a genital tract infection, the cloaca being the combined anal and urinary urinogenital opening was sometimes inflamed. So here's a severely affected young one. See that the eyes are virtually completely closed. This white material is what I'm calling fibrin, discharge, high protein sort of discharge, and at the back of the mouth severe build-up of the same sort of material. Eyelids are swollen, the eyes are often closed, they're often blind and can't see their food. Some animals presented at what appears to be an early stage where they're just very red at the back of the mouth and probably a bit eroded. And then most of the animals had a more advanced stage where they had this build-up of this white fibrin-like material. And the important thing here is over the back of the throat, this little area here is a slit through, it's the actual laryngeal opening, and that quite often was blocked, and that was the reason for the majority of deaths in these young animals, is just a build-up of this fibrin-type material, just as diphtheria used to happen in children back in the earlier in the last century. They just couldn't breathe and would die, and that's a really advanced case with massive build-up of the fibrin. This is this cloacal area, the urogenital and anal opening, and occasional animals had the same sort of fibrin-type material occurring there. In the older animals, I said the other farms started hunting around to see if they could be at the early stages of an outbreak, and the two other farms for some reason didn't have any disease in their hatchlings, but they found occasional older animals with similar lesions, and we thought these could possibly be a sort of a carrier group that may be infecting the younger ones. That was the theory. So these older animals had a thickened third eyelid that comes across. Normally that's pretty clear, and this white material. When those ones with those eye lesions, when you looked in their mouth, often they had, at the same time, they had severe lesions at the back of the throat. What we saw down the microscope, the normal lining of the inside of the eyelid is these epithelial cells, which are long cells like a picket fence extending out that way, with a nucleus at the bottom, and this is all the cytoplasm of the cell. and clean underneath that lining, clean connective tissue underneath. In these affected animals, those cells were basically lost and replaced by stunted, flattened cells that were just trying to cover the surface, and underneath the surface a lot of inflammatory cells, lymphocytes and plasma cells coming in. Same in the pharynx. These picket fence type cells, lubricating type cells that line the pharynx, were converted to these flattened, stunted cells and severe inflammatory cell inflammation, whereas down here it's quite clear normal tissue in the normal animals. So that combination of signs and lesions, although the lesions are not specific, but they are things that we've seen before with chlamydial infection in other species, koalas for example, humans get a disease trachoma, and lambs get pink eye, and these are all chlamydial diseases. So it was one thing to target straight away. There were anecdotal, or I guess there were some even textbook reports of chlamydia causing conjunctivitis in crocodiles. There's been no published material on the conjunctivitis caused by chlamydia in crocodiles. So we're sort of breaking new ground in a way, and we sought these chlamydial PCR tests done at the Townsville lab, DPI lab in Townsville, because we hadn't had a chlamydial PCR up until now at our lab. And we sent off 14 samples of conjunctiva from 14 animals, and they're all positive for chlamydia, and 6 pharyngeal samples from 6 animals were also all positive. We do have an older capture ELISA based test at the lab, and that was tested initially and gave fairly weak reactions in 3 out of 6, which subsequently turned out to be all positive by PCR. So this is a considerably lower sensitivity test, as we know from other comparisons of PCR with capture ELISAs. We look for other agents as well. At the same time, we sent off specimens again to Townsville lab because they had a mycoplasma PCR as well. Only 1 of 6 samples came back positive for mycoplasma, and none of the 4 pharyngeal. That was 1 of 6 conjunctivas, and none of the 4 pharynx samples were positive. Richard Weir looked at some thin section electron microscopy using Charles Darwin's University's EM, and none of the 3 animals we looked at, that Richard looked at, had viral particles, and none in the pharynx had viral particles in the pharyngeal tissues. We did hope to culture for viruses, and we attempted to import a cell line. There's no reptilian viral culture cell lines in Australia being run at present, and we attempted to get there's no crocodile cell lines available or alligator cell lines, but we did attempt to get a turtle box heart cell line in, and that kept on being from the US, and the necessary permits weren't being issued, and eventually after several months or almost a year, I think, Richard, the US authorities decided they weren't going to issue any permits, and it all relates back to this box turtle was an endangered species, and you're working from a cell line from it, and you're not working with the turtles, but they still banned export of that cell line because it was an endangered species. Anyway, just to outline what chlamydia are and how they develop, I've put up these couple of shots. Chlamydia are actually a bacteria. They're originally or previously classified as rickettsia, but they're a bacteria, and they're different to most bacteria in that they need cells to reproduce or replicate. Most other bacteria can replicate just within body fluids or within the environment. Chlamydia need the living host that can't replicate in the environment. And what happens is these infectious particles, called elementary bodies of chlamydia, that stage gets into the host cell. It creates the formation of a sac, which we call an inclusion. This is the sac here, and the particles start budding off, reproducing from the lining of that sac. Eventually that sac forms the whole cell. The initial ones are a little bit larger. These are about 1 micron diameter, and as they mature they condense down to the infectious type particle, which is only about 1/3 of a micron in diameter, those ones there. And then eventually that sac fills the whole cell, and the cell ruptures and releases those to infect other cells in the eye or the pharynx and to infect the environment. As I mentioned before, chlamydia cause disease in a range of species, and this is the classification of the family Chlamydiaceae. Two actual genera in the family, one chlamydia, which has got the trichomonas, which causes the most common infectious cause of infertility in women, most common venereal disease in humans, and a couple of other ones that occur in animals, not really of much significance, but they are used as a model for the human infection. Most of the veterinary the ones of veterinary significance are in this Chlamydophila genus, and the most commonly known one is psittaci, causes psittacosis in birds and causes severe lesions in humans. If you get exposed, a lot of veterinary pathologists have got psittacosis in the past, and it can be fatal. It's quite treatable with certain antibiotics. Other ones, abortus appears to have evolved from psittaci. It's very close, and that's a significant cause of abortion in sheep in other countries, not in Australia. Caviae in guinea pigs causes conjunctivitis and infertility, felis in cats, pneumoniae in a whole range of species, including humans. And pecorum is the one you might have seen a lot of publicity about, koala disease, koala conjunctivitis, koala wet tail, which is infertility in koalas. That's pecorum that does that. Okay, so where's the crocodile fit in? Where might it have come from? Has it come from birds? Has it come from amphibians, or is it its own type, unique type and species? And the way to sort that out for the other species, and the way we wanted to tackle the type from crocodiles, is to initially perform the PCR test to amplify the specific DNA belonging to that family. Once that's amplified, we either sequence that segment of DNA to find the specific sequence for the species and the strain, or we do RFLP, restriction fragment length polymorphism, which is using some enzymes to chop up the DNA. The set of pieces, the particular combination of the pieces when you chop up that DNA classifies it into one of the 9 species, hopefully. Oh, okay. So the genes that you might use for doing this sequencing to place this chlamydia in a species, there's a couple of ribosomal genes, and that's how that structure, that chart I gave before with the 9 species, they were divided up into those species based on the combination of these 2 genes and the sequencing. The one we're particularly interested in is called the OMP2 gene, OMP being the outer membrane protein, codes for a part of the actual outer membrane of the chlamydia. This one's of interest to us because we can use it on clinical specimens. You don't have to culture the organism first. Use it on clinical specimens because it's quite specific to chlamydia, and then you can go on and do this RFLP rather than going through the whole process of sequencing to try and get a species. And this is this RFLP technique, this enzyme digestion of the DNA to break it up into a species. So what we've got here is this is the raw bit of material, undigested, 1,030 base pair units long. This is a marker in this column that's been digested to a number of fragments of known lengths. And these are the 9 different species. The other 9 columns are the other 9 different species of known of chlamydia, and they all have their own particular pattern there or fingerprint that identifies that species. So researching into chlamydial infection, because that was there in all the animals, there wasn't any other potential pathogen identified, although we hadn't fully investigated the viral story, but it was really the only one that was there that was looking like a prime candidate. We decided that it was worth applying for research funds to answer a number of questions about this disease. Where is the reservoir for this chlamydial infection? Is it right through the population? Is it in just young adults, or is it in just the very young hatchlings and then they get over it? Is it more common in poor, ill, thrifty animals than healthy animals? This will all help to understand the role of chlamydia in this disease and in other possible conditions within crocodiles. What species or strains of chlamydia are present, and have these come from somewhere else, or are they unique to the crocodile population? Is the infection common in the wild population? Could it have suddenly come into these farms last year from the wild crocodile population? So they're all the questions. And could it have come in the eggs, or has it come in from a water source that's been introduced? So we applied for funds and were successful, and we've got a one-year project. As I mentioned, we're a few months into that project. The project was designed to have two stages. One was to introduce the technology to the vet lab, and this was the PCR testing method, followed by the enzyme digestion method, the RFLP, and sequencing to speciate it. And we proposed that it would be worth looking at these three genes, the two that the whole system's been based on of classification, but this extra one that might allow us to do a shortcut to identifying the chlamydia through this faster RFLP method of speciating. As part of that, as part of the work up to these molecular methods, we didn't want to just work with clinical specimens that got a whole range of DNA in them. We wanted to work with chlamydial DNA, and so what I organised was the import of reference strains for as many species as we could get of chlamydia, imported from the US because there's no Australian reference collection for chlamydia. So we had to go to the ATCC, the American type collection, and import those at a cost of about $800 per strain to get them here. And so we were hoping we would be able to successfully culture them, and Richard and Christine have successfully cultured the first two strains that we've put up. Growing in these flat bottom flasks, they form a monolayer on the flask, and once they're at sorry, these are HeLa cells growing in the flask, the actual cell cultures, and once they've formed a monolayer, the chlamydial material, the reference strains, are inoculated into those, and that allows us to grow up the chlamydia to give Lucy and others working in the molecular area a pure sample of chlamydia to work with and evaluate our methods. And the way to evaluate whether chlamydia growing in those cell cultures, whether it's successful or not, is to look at immunofluorescence on the cells that are growing in there to see whether they're full of chlamydial particles. And this is not our photos, this is a download off the internet, but these are cells infected with chlamydial particles. The slightly larger ring-like ones are the early stage, the reticulate bodies, and when they condense down to the more infectious particles, they're the solid little particles here. The background cells in ours, the background cells that are not infected are stained a reddish brown because we have a Evans blue dye, and there is a counter stain. This is not the same method that we use, but in this method these are background cells that are not infected, no fluorescence of any significance. So the aim is to grow the chlamydia to harvest for reference DNA for us to store, to validate our methods, and to attempt to culture this crocodile strain so we can fully characterise it. Once we get it in pure culture, we can sequence it and fully characterise it and report it to the world, I guess, as to what this finally is. Because crocodile chlamydial infections have been talked about for years and years, but no one's fully characterised the strain. And it'll allow us to evaluate antimicrobial resistance. Now, tetracyclines have been the mainstay of treatment of chlamydial infections. There are newer drugs now, like azithromycin, that work very well because they last a long time in the body. One tablet lasts a week. But there is some concern that the chlamydia in the crocodiles, being an intensive farming operation where they're loading lots of tetracyclines into the farming environment, that there may be resistance there. So growing it will allow us to evaluate that. It's not part of the project, but it's something we can do in the future. All right, so this is what we're just covering back what I said before. There's three genes that we're interested in. Lucy's done some work now. And got that OMP2 PCR working on the reference strains, getting nice clear bands, and we had these. The 16S is another one that Lucy's been working on, and it's proved not good on clinical specimens, but works well on the specimens in culture. The second stage of the project is to sample the population. Groups ranging from hatching up to 18 months old, 700 animals in total, looking at some groups of runt animals, looking at eggs collected in the wild, and looking at crocodiles trapped in the wild. Already NRETA has given us cloacal and eye swabs from 60 crocodiles that were trapped for relocation to the croc farms. Eggs will be collected this season, starting in December. We've sampled young animals from, I think we've got sampled about 240 young animals so far, and we've sampled some of the runts. And this is the type of sampling we do. We use a fine aluminium shafted swab to get in under the eyelid and sample the pharynx as well. We've even sampled some animals basically as they're emerging from the eggs and in the first week of life. Some of the findings that have come through from other things, that in the meantime, prior to us getting our methods established, we'd sent stuff off to the University of Sydney, who've got the big project on koala diseases and have got their methods set up for that. And we did get the 16S sequenced on strains from 2 animals, and that's the sequence. The alignment of it, Lucy confirmed Tiziano's from the University of Sydney's alignment results, which show that the Crocs strain from the 2 animals that they tested there aligned most closely with the Psittaci, and we would expect that because reptiles and birds, and Psittaci is a bird strain, have the same sort of ancestry. So we would expect it to be close to that. But it also was very close to the abortus. This is the strain that was probably evolved from Psittaci, so expect to be around that area. And it was also rather close to this other ovine strain, which is probably just a subgroup of abortus. So it didn't fit in classically into any of those strains. Now that was looking at the 16S, the way the whole system's been classified in the past. We also, Tiziano also did sequencing on the OMP2 gene, the newer one that we want to use now, and that had a different alignment. It was closest to the guinea pig strain, but it was also close to Psittaci. So this speciation is proving to be quite difficult. It depends on which gene you use as to which species it's closest to, but it doesn't classically fit into any of the 9 species. I think that's the key, that this is likely to be something that's unique to crocodiles. And we did sample herons. During that project there was a lot of publicity that herons had brought the infection to the farms from the tip, and we did sample some herons and send them to University of Melbourne. They matched 100% with Psittaci. They were classic Psittaci from the herons, and they were clearly different from the crocodile strain that had this some sort of cross-reactivity with caviar. Okay, so that's where our crocodile strain fits in on the chart, fairly close, probably closest to Psittaci and abortus, and also with some sort of similarity to caviar, but clearly away from this pneumoniae and clearly away from the one that causes disease in koalas and cattle and sheep. The heron strain fitted classically into the Psittaci, the other birds, where the other bird strains are. Some other work that was done for me by Damien Higgins, at the University of Sydney again. It was immunoperoxidase, looking for the chlamydia in the tissues using immunological staining, and that was quite successful in demonstrating the chlamydia in the tissues in substantial numbers, but unfortunately not at the sites that we were expecting it to be at. So that's a positive in the lung, and that's a control negative from some different animals. I submitted some older animals that came in without any mucosal lesions. 3 out of 3 of those were negative. 5 out of 6 of the hatchlings with the eye and throat lesions had chlamydia in substantial numbers that you could stain up in their tissues. But unfortunately, most of the staining was in the liver and lung, with only one of them having stainable material in the pharynx. This is the pharynx here, and there's no significant staining in the majority of animals. So that was a bit of a letdown, I suppose, to the fact that we couldn't prove that chlamydia were causing these lesions in the eye and pharynx because we couldn't locate them in substantial numbers in the tissues there. But talking to the researchers in koalas, they find that's not uncommon in the koalas. The damage is done often by the time you get these sections at the site. Most of the damage that's occurring is the body's immune response without the organisms there at the time. So we're still up in the air of that. There's that association that chlamydia there in the tissues of these animals, but unfortunately not for us, not located at the site that we expected. Other results that we've got so far. We detected positives in normal hatchlings as they're emerging from the egg. Now, we don't know whether those eggs have been infected at the hatcheries. The actual procedure with crocodiles is the eggs are actually 80% of the eggs, 90% of the eggs are collected in the wild, and they're incubated at the properties, hatched at the properties, and then raised there. But these ones emerging from the eggs, they could have been infected in the incubator, so we don't know the significance of that. And we have detected quite a few positives in ones up to 7 days old. And detected the organism also in a group of runt animals. We haven't done many animals at this stage, and all the testing's been done in Townsville because we've just been concentrating on working our methods. But they're the results, early results so far. Early conclusions are mucosal chlamydial infection, that's of the pharynx and eye. It's common in diseased, and it's also present in clinically normal crocodiles. The types, the phylotypes of the chlamydia are closely related to psittaci and abortus, but are probably represent a novel species unique to crocodiles. The relationship of the mucosal chlamydial infection to disease is uncertain because we weren't able to prove by locating the chlamydia in those tissues at the time of getting the samples that it was the causative agent. So that's where we're at to date. And that's the acknowledgements. This group, quite a large group involved in the diagnostic workup to this disease. A lot of help from interstate labs, particularly Townsville, University of Sydney, University of Melbourne helped out. The funding bodies, rural